Great pleasure to be able to introduce Kamil Ugurbil from the University of Minnesota. Kamil holds a PhD in physics and chemical physics from Columbia University. He received his PhD and then joined AT&T Bell Laboratories and subsequently returning to Columbia in 1979 as a faculty member. In 1982, he moved to the University of Minnesota, where he was a where his research effort in magnetic resonance led to the evolution of his laboratory into an interdepartmental and interdisciplinary research center called the Center for Magnetic Resonance Research. Dr. Ugurbil currently holds the McKnight Presidential Endowed Chair Fellowship in Radiology, Neurosciences, and Medicine, and is the Director of MR Research at the University of Minnesota. His research focus has been in the development of biological magnetic resonance imaging and spectroscopy using very high magnetic fields with a particular emphasis on brain function, anatomy, and chemistry. One of the two studies that introduced functional imaging in the brain using MRI was conducted at Minnesota under his leadership. Since then, his work has primarily revolved around the understanding the origins of MR-detected functional signals and developing strategies to improve the spatial accuracy and spatial resolution of the functional maps obtained by MRI. The use of ultra-high magnetic fields, that is, those that are 7 Tesla and higher, for human studies was pioneered by Dr. Ugurbil as part of this neuroimaging effort. Professor Ugurbil's contributions to biomedical magnetic research were recognized with a gold medal by the International Society of Magnetic Resonance in Medicine, the ISMRM, in 1996, which is the highest award given by the society. He was subsequently elected to a fellow of the ISMRM in 1997 and of the International Society of Magnetic Resonance in 2009. Dr. Ugurbil was inducted into the American Academy of Arts and Sciences and the National Academy of Sciences Institute of Medicine in 2005 and 2007, respectively. It is with great pleasure to introduce Dr. Ugurbil. Thank you.
Thank you, Andrew, for that introduction, and thank you very much for the invitation to come and present this lecture to you today. I wonder if we can have the lights down a little bit, because I will be showing quite a bit of images. Naturally, it's a talk on imaging, and bright lights don't go well with imaging slides. Well, you have read my title. It is my title is Frontiers in Neuroimaging, from cortical columns to whole brain function, connectivity, and morphology. I've added actually to my title a revolution in imaging, because I think that what has transpired in the last 2 decades in imaging of human brain function, activity, and morphology is really can be characterized as a revolution, and a revolution that is still going on. Approximately 2 decades ago, this was the state of art in terms of functional imaging or metabolic imaging. The technology was based on positron emission tomography, and here you can see actually images depicted for visual stimulation in the human brain. And the images outline the visual cortex in the human brain right over here, and you can actually see activity there. Should I speak that? You can see activity in the visual cortex, but not a lot of great detail. Maps contain of blood flow, glucose utilization, oxygen utilization, and oxygen availability are, by today's standards, extremely poor resolution, and in some ways quite inadequate for the task at hand. On the other hand, I have to say, for those two decades ago, this was indeed a fantastic achievement, and but has been surpassed since then. Today, we can obtain anatomical images of exquisite detail with magnetic resonance imaging, as shown here from 9.4 Tesla images of the human brain. We can follow connections in the human brain of major axonal bundles and from various different regions. We can, of course, map activity in the brain with high resolution. The resolution can go from millimeters. For example, now we are talking about visual stimulation here, just like the PET study, except that we can see pathways like lateral geniculate nucleus, primary visual cortex, which can be subdivided very easily, and all the way down to sub-millimeter visual organizations. And we can also map metabolic processes, such as, for example, rate of various different reactions. On top, you see the rate of creatine kinase activity in the brain as a function of brain activity, and at the bottom, you see oxygen consumption rate. So all of these developments have come as a result of significant efforts undertaken for instrumentation, and that instrumentation includes the use of ultra-high magnetic fields. The picture of the 7 Tesla magnet, for example, going into our lab in Minnesota in 1999. And in addition, multi-channel receive arrays and multi-channel transmit technology. With respect to the high magnetic fields, I have to say that this has been a crucial development. And the first 7 Tesla system we established in 1999 in Minnesota. This was actually a homemade system. And soon thereafter, now one can buy these systems now commercially, which has led to this explosive interest in the establishment of these systems as the major research instruments having to do for brain research. And in addition to instrumentation, of course, there has been incessant, I would say, that's a good characterization, incessant discoveries and improvements in methods of image acquisition and reconstruction that has fueled this development. These developments really were necessary for us to reach different operative scales of the brain, from cortical columns to networks, and we had to reach this to be effective in the whole brain. We cannot access, by magnetic resonance imaging technology in the human brain, for example, individual neurons. They are beyond our ability to resolve. We cannot see all the synaptic connections these individual neurons make, and make circuit diagrams using all of the individual synaptic connections. However, there are other scales, important scales that we can reach. For example, cortical columns. They are composed of something like few thousands of neurons, and they can be regarded as elementary computational units, and they perform a fundamental and a simple process. Neurons are segregated across the cortical layer. So, for example, if this is the top of the gray matter ribbon and this is the white matter, 6 different layers of neurons with different characteristics and functional properties can be identified. So we can access cortical layers today with magnetic resonance technology. And of course circuits, but not microscopic circuits at the level of individual neurons, but circuits of functional units, such as this diagram that I have shown here, where we are talking about entities which may spatially occupy several millimeters to centimeters of cortical space. Interestingly, while we cannot access the individual neurons, magnetic resonance technology can actually access the chemical processes that go on in the synapse through magnetic resonance spectroscopy. But this is a different topic that I will not delve into today. I will speak mainly of the imaging efforts, and would like to show you that indeed we can go from cortical columns to networks with great success in the human brain at the present time. A very good example of cortical columns is the ocular dominance and orientation columns in the primary visual cortex. Here you have the output from the 2 individual eyes segregated already very early on in the lateral geniculate nucleus to different populations of neurons that actually respond to the 2 different eyes. When you go to the primary visual cortex from these lateral geniculate nucleus, then again you have these sort of cartoon-like depiction of columns which are representing different populations of neurons segregated in space, kind of like columns, and performing calculations on the two different eyes. And on top of that, columnar organization, the so-called ocular dominance columns, it is well known, for example, there is representation of orientation. This is in the visual cortex, where the orientation in our world, what we see, is represented in a distribution of 360 degrees around singularities, what is normally called pinpoints, singularities in the cortex. So these orientation domains can be mapped as well as ocular dominance columns in the human brain for the first time recently using magnetic resonance imaging techniques and high magnetic fields. So this is from our work in 2007 showing you ocular dominance columns. And the imaging was, of course, performed in a rather large section of the brain and using a single slice. But we are just looking at this area over here in the primary visual area where the cortex is in this individual is flat, and we can actually see the layout of the ocular dominance columns very nicely. So this is the image from this area showing one eye versus the other eye. The spatial scale here is approximately 1 millimeter. That is the size of the ocular dominance columns. And they represent indeed a clusters of neurons that process input from one eye versus the other eye. The orientation domains are also mapped in 2008. We reported this using, again, functional imaging and ultra-high magnetic fields, in other words, 7 Tesla, in the same individual. And we can even then compare and contrast the organizational characteristics of the two columnar systems. So you can see the outline of the ocular dominance columns from this image over here in black, and then the orientation representation of orientation over here in color-coded superimposed on that. With this work, we have been able to, in fact, today go from mapping orientation domains in the animal systems like the monkey with optical imaging techniques for the first time to humans, and with using functional imaging techniques and ultra-high fields. And they look very similar, and the characteristics are similar, but indeed human ocular orientation domains had never been mapped before the study with functional imaging. This was a study where we had used only one slice, looking at not the whole brain, but just the visual, primary visual cortex in a particular individual where the cortical architecture was flat so that we can visualize these without having to deal with the complexity of the curvature of the human cortex. However, recently we have extended that to imaging in fully in 3 dimensions, but in a different columnar organization. This is mapping motion direction selectivity in the human area MT. Human area MT is also part of the visual system, but it's a much more advanced area in the visual system as opposed to the primary visual areas. In this area, there is also direction sensitivity, but in fact, the orientation sensitivity, but it's actually more than that, it is direction sensitivity, as I will show you. This work was conducted with our collaborators from University of Maastricht, and the data was again acquired at 7 Tesla. But now we are talking about volumetric imaging as opposed to single slice imaging. The paradigm is something as shown over here. You can see that this is a dot pattern, and they have direction movement in a single direction as opposed to the orientation I showed you before. The human area MT can be identified with various different paradigms, which I don't have time to show, but it is actually located laterally, as shown here for one of the hemispheres. And it is a curved surface, and it is very difficult to image that on a single slice. But with now volumetric techniques possible, with the kind of resolution here shown, 0.8 millimeter isotropic, for functional imaging. This is not structural imaging, so that this image has to be acquired relatively fast, and it has to be repeated one after another. And that in that fast acquisition, which takes approximately 100 milliseconds or 200 milliseconds, there is actually the ability to obtain the functional signals with this resolution at high fields. And it's a curved surface, and we have to use data processing to segment this into different layers and then take care of the curvature. So we actually segmented this into 3 different layers shown in blue, red, and green. And but it is actually what you are seeing over here is a 3-dimensional structure with a curvature that is being projected onto a plane. And here's the sulcus, that's the dark area right in the middle. And here's the gray matter area, the cortex over here, which is approximately 2 to 3 millimeters thick. And at this resolution, then you can map the columnar organization of direction selectivity, but with, in fact, specificity to different layers of the cortex. So it is right here shown for the MT area. These are the superficial areas, middle area, and the deep areas. So we indeed can look and achieve imaging of columnar organizations, and as I had mentioned, an elementary computational unit in the human brain using high-field functional imaging these days, and achieve it with differentiation of cortical layers. These are the tuning curves that are obtained from that. These are tuned clusters, tuned columns, tuned clusters of cells. And for each orientation, you can see that they have a relatively narrow response. There are 2 different curves shown here, gradient echo and spin echo. These are 2 different imaging methods. I won't go into the detail. Only for the imaging aficionados, this is of interest. But the important point is actually that both of those techniques give you the ability to detect the tuned response of the brain. This type of imaging is very important and necessary, in fact, if we are going to make a connection between input and behavior versus neuronal activity. And then the connection these days can be established by various popular methods. And for example, decoding is extremely a popular method. It's sometimes referred to as brain reading. It's based on classification algorithms, and it is model-free. It doesn't necessarily tell you very much about how the brain works, but it does give you essentially a signature of brain activity associated with certain behavior or input. And people have been successful in, for example, being able to figure out what pictures people are looking at, and we have been able to reconstruct the sounds that you hear in a magnet, that you visualize that you hear in a magnet in the human brain. So this is decoding. It's a powerful technique that is employed quite extensively to understand brain function. And there's a variant of this, which often referred to as encoding. It is really a special case of decoding, except that it has a neural model in between. So in this case, you have a little bit more information, or you require information, or you can build more information about how the brain works, whereas just pure decoding doesn't care how the brain works. In the encoding, it's actually a decoding with a model. And in these 2 specific strategies, which are becoming very popular and I think extremely promising, especially the encoding approach in terms of understanding how the brain works, ability to obtain these very high-resolution images with information-rich voxels is very important. Let me tell you a little bit more about that using the columnar organization that I just showed you. For example, we talked about the ocular dominance columns, and in our data, they are very high resolution, but a high resolution image can always be processed as a low resolution image. So we were able to reprocess the data or bin it into a low resolution bins, for example, 2 by 2 millimeter or 1 by 1 millimeter, as opposed to 0.5 by 0.5, which is the native voxel. So in the classification algorithm, what you can do is you can take functional imaging data. For example, functional imaging data essentially is data that is repeated one after another, and then somewhere in between there is some sort of a stimulus that comes in. And these images you acquire it as a time series. So you can divide that into 2 and use one half of that data set to train a classifier. And subsequently, you can use that classifier to examine the second half of the data that was not used for the training. So the question is, if you choose one of those images from the time series, can the classifier identify it as left or right eye stimulation? And this would be a decoding algorithm. And indeed, you can successfully do so. And for example, this is the performance of a classifier looking at the spin echo data for ocular dominance columns as a function of the voxel, as a function of the volume that you allow the classifier to see and examine. So when you obtain data at these various different resolutions, and you look at a volume of for example, approximately 200 cubic millimeter, the classifier tends to fail at these very low resolutions, but tends to work pretty well at these somewhat higher resolutions. But what is really interesting about this data is that when you go to, oh, sorry, I went backwards. When you go to very small volumes that the classifier looks at, that very high resolution data, such as the one that is at 0.25 by 0.25 millimeter or 0.5 by 0.5 millimeter, it still works very successfully in the classification. So now you are looking at only 12 cubic millimeter of cortex, and you can actually identify from this data whether that one single image you pulled out from the time series is a left eye or a right eye stimulation. And this is not only a testimony to the robustness of these data in terms of their information content, but it's also a testimony to the fact that when you are using decoding algorithms, that you need richer information content of the brain, which is provided by these high-resolution images, and higher contrast and noise ratio that comes from ultra-high magnetic fields, and a large number of informative voxels, which is provided when you actually go to these high resolutions. I have to say that when you actually perform these experiments, if you were to perform these experiments at this kind of resolution, at lower fields, they may not be as successful because of the fact that the point spread function of the signals, functional signals, is quite a bit more extensive, and then independent and informative voxels are not as large. This type of imaging has become very important recently in the so-called Human Connectome project. This is an initiative that was funded by NIH, and the goal is to chart the macro connectome and its variability in health and healthy adults. So there are two consortiums involved in this project. One is the so-called Washington or WashU University of Minnesota consortium, which is the bigger effort in terms of mapping this. The second one is a smaller effort from MGH and UCLA consortium. So in our consortium, there are 9 institutions led by Washington University and University of Minnesota, but in participation with several other universities, including Oxford, Indiana, and others. And the idea is to map, to generate a data set, a comprehensive map of neuronal corrections in the brain. But I have to immediately clarify that we are again not talking about microscopic connection, not talking about a microscopic connectome where one has to map synapses between neurons. This is an effort that that does go on in small animals, including mice, but we're talking about a macroscopic connectome and between regions of the human brain, but in the entire brain. There are several different MR technologies that play a role in this effort, and MR is the primary technology that is used for this effort. One technology is diffusion-based tractography, which comes in several different flavors, such as high angular resolution diffusion imaging and diffusion spectroscopic imaging. But there are other methods that are used: resting state fMRI, which I will talk about, and task-based fMRI, which already I talked about a little bit in terms of columnar imaging. And task fMRI is used for functional parcellation, and resting state fMRI is a tool for functional networks. And each of these, when you have to deal with whole brain acquisitions at high resolution, faces a challenge. And the challenge is if you are trying to do diffusion-based tractography, for example, and you want to acquire enough data so that you can actually untangle fiber tracks that cross each other, then you may be talking about acquisitions which last 40 minutes or so. Of course, rather unpractical data acquisition time for a database which will have many, many individuals in it. On the other hand, resting state fMRI and task fMRI, the challenge is, again, it takes too long. It's a different spatial, it's a different scale. It's not 40 minutes we are talking about, but we have to acquire in seconds and ideally sub-seconds, but the imaging, when you are talking about high resolution, is rather long as well. So if you look at, for example, 7 Tesla single-shot echoplanar imaging, for those of you who are not very familiar with MR methodology, that is essentially you excite the spins and in one shot you acquire multiple slices, and it takes you of the order of 50 milliseconds or so to collect a single slice. And then you repeat that for each slice. So as a result, to cover a whole brain image like this, it takes approximately 6 seconds. So 6 seconds, then you would have to repeat that every 6 seconds to get an fMRI time series, whether you are talking about a resting state fMRI or task-based fMRI. Of course, you may say, well, 6 seconds with 0.75 millimeter isotropic resolution is not bad. It's actually pretty damn good, but it is still actually rather long. And we have tackled that problem when we had the ability to get to those resolutions at 7 Tesla. We had to tackle that problem, and we tackled that some time ago and made significant improvements in that. So in conventional multi-slice imaging, as I have indicated, you acquire one slice at a time. A single slice may take 50 or 60 milliseconds or so to acquire, but then if you are covering it at high resolution, you may have to acquire 150 slices or 170 slices to cover the whole brain when you are dealing with high resolution. And it takes then, the time of acquisition then goes linearly with the time of, the number of slices that you acquire and the time per slice. On the other hand, you can, instead of doing that, you can obtain an image by exciting multiple slices at the same time. For example, you can excite 4 slices and then march 4 slices at a time, cutting the acquisition time by 4. But when you acquire 4 slices at the same time, then you have to somehow untangle those slices because the image now is a sum of those 4 slices and it's a blurred image. And you use then coil sensitivities to do that. Today, it is rather standard to use multi-channel array coils to acquire data. And in multi-channel array coil, there is spatial information. It is used in conventional imaging in a different way, for example, for parallel imaging along phase encoding direction. But in this case, we have used it to untangle slices. So, for example, you can look at a 4-slice set like this one here in red, shown here, and say, how can we untangle that when we acquire them simultaneously? Well, this particular one here is, you can see that it is really very highly sensitive to these coils. These coils are very highly sensitive to this particular slice. On the other hand, the coils over here are very sensitive to that slice. And you can actually find a coil combination that are uniquely sensitive to all of these slices. And each coil, in fact, has a weighted average or detection for each of these slices. And you can perform a matrix inversion to actually untangle those. And today, you can, in fact, perform that with a lot of improvements that came up subsequent to the fact that we performed it in 2008, improvements that came from MGH, and then some subsequent improvements from our lab. Today, you can, in fact, acquire, for example, this is a single slice normal imaging, just showing you 3 of the slices from the brain. And these are 2 slices acquired simultaneously, 4 slices acquired, 8 slices, and even the 8th slice looks very good. And 12 slices, it starts getting a little bit blurry at this point. And these imaging, the number of slices you can acquire, also depends on the number of elements you have in a coil array, and can be improved again using hardware improvements that are, I'm sure, on the way. So with this kind of a technique, now we're talking about sub-second whole brain images, which are of relatively high resolution. And in this case, you'll see a 3 Tesla image. We are working also at 3 Tesla for this human Connectome project. It has actually 2 components, a 3 Tesla and a 7 Tesla component. And you see 3 Tesla images of 2 millimeter isotropic obtained using 6 simultaneously excited slices and in sub-seconds. We recently further accelerated this. For example, this is a pulse sequence for a single EPI image. You don't have to be an MR expert to understand what I have to tell you. The important point is you excite and then you detect. And then you do spatial encoding over here. And when you do these multi-banded pulses, the only thing you do is you change the excitation pulse to a multi-banded pulse that excites simultaneous slices. And you subsequently, of course, have to do a rather complex image reconstruction. On the other hand, there was another technique that was published some time ago called SIR. And in this technique, you try to acquire multiple slices, but you don't excite them simultaneously, but rather separated from each other in time. There are some limitations here. You cannot really go to much more than 2 or 3, depending on the field strengths and the gradient strengths you have. But clearly, these two things are complementary and can be combined. So you can combine the SIR with the multiband, and then each pulse is a multiband pulse now. For example, if you use a multibanded pulse of 6, and then you are collecting 3 excitations acquired subsequently in time, and then you end up with a 3 × 6 acceleration. So for example, here are some images from this regular EPI, and then the so-called SIR-MB combination, which gets up to very high accelerations. You do lose some, you do get some distortions when you go to this very high SIR factors, but there are also ways of dealing with that as well. We have demonstrated that these high accelerations is very important for these kinds of network studies. For example, let me talk and introduce you to this concept of resting state networks, which are dealing with functional connectivity. There are spatial patterns of correlated temporal dynamics in the brain that are detected in an fMRI time series, and they resemble activation maps as shown over here. They can be found in fMRI data, or they can also be found in MEG data. And the idea is, in fMRI, as I had shown you, we are acquiring one image after another in a time series for a very long time. So when we talk about a resting state network, we're of course not talking about the brain resting. The brain is not resting, but the subject is just resting in the magnet, doing nothing. You can collect these data with eyes closed or eyes open, but no particular task or stimulus. And if you look at different areas, for example, in this plot here, this is a time course of data from the brain of 4 different regions in different colors. And you can see that even all of the colors are more or less going up and down somewhat similarly. So they are highly correlated. And when you map those correlations, you get these resting state networks. And the idea is that there is some sort of a functional connectivity between those regions. And the kind of things that we can do with these fast acquisition is actually, it turns out, very important for these resting state networks. So here's a data obtained. This was published recently in PLOS ONE, obtained with a regular acquisition time. This is a 3 millimeter isotropic resolution. So that means you can achieve TRs like 2.5 seconds. And you can detect a resting state network in this particular slice of something like this over here. So these are correlated areas in the time series. On the other hand, when you are able to accelerate your acquisition so that you can now sample whole brain data in 0.8 or 0.4 seconds, then you can see that the areas that we detect are much more significant. The color code has gone up from red to yellow, as well as somewhat larger. and you can quantify that using statistics. And these are ICA analysis, independent component analysis, and this was a 100-component independent component analysis, and each of these components, when you look at the statistical significance of those resting state networks, here is the normal acquisition, 2.5 seconds. Here are the shorter TR acquisitions. And you can see that the 0.4 second TR tends to be somewhat better than the 0.8 second TR in many of those components, but not all of them. And both of these 0.4 and 0.8 second TRs better than the slow acquisition. So indeed, to capture those resting state networks, we are interested in these rapid acquisition schemes of the whole brain. But this is, I regard this as an incremental improvement, however. And on the other hand, these rapid acquisitions, in fact, has allowed us to look at some new concepts. For example, this concept of transient co-activating networks. So resting state networks are based on assumption of stationarity, and correlations are average and not changing over time. But this is a simplified assumption, I'm sure, for the brain is not very stationary. So the aim was to identify essentially transient networks that may be coming and going in the brain at different times. And this is work, I should say, the data, all the data comes from Minnesota with these techniques, and the analysis is by Steve Smith at Oxford University. And so you may be, so the aim was to look at these dynamic activity, and then here then we require many, many more time points, which is then facilitated by these very fast whole brain acquisitions. So if you were to look at a brain network like this schematically, let's say that it has 5 nodes and edges, connections between them, you may or look at 2 different networks that are identified, something like this over here, and you may have actually a temporal correlation between these all the time, so you may have a transient co-activating network like this and like that. But occasionally, you may have a correlation between these nodes that are transient and take place briefly, and then you go back to others. And indeed, this happens, and I can show them to you in a movie. Here is a movie of 21 transient co-activating networks that come and go, and the movie is approximately 6 times faster than the real time that is taking place in the brain. And you can see each of these multiple areas that are activated and they come and go. This is essentially a network with many nodes. And they are correlated at that period. We call them transient co-activating networks. And they are not stationary, and they are changing with time. If you look at it in a more static fashion, for example, this is one of those such networks. The visual areas in the brain are anti-correlated with areas on the temporal lobe, which is part of so-called the default network. So there is still quite a bit of new work to be done here with the transient networks, but on the other hand, the technological developments recently driven by the Human Connectome project has already allowed us to look at new insights or new processes in the human brain. Some of the networks that I've shown you were at 3 Tesla, as I had mentioned, because of the fact that 3 Tesla is part of the Human Connectome project, but so is 7 Tesla. And at 3 Tesla, we identify networks with relatively low resolution, but as you can see, at 7 Tesla, these networks become identifiable with very high resolution. And now you can actually see them very nicely corresponding to cortical gray matter areas. And of course, they are whole brain images, just to show you that I showed you just one of the slices for different networks. But they are whole brain images. They cover the whole brain, and with these methods. And you can go to, for example, 1 millimeter isotropic, which is really a very high resolution for these resting state networks, and I think probably somewhat higher is feasible. And the important part here is that, for example, if you look at the sulcus and the activity on one part of the sulcus versus the other part, this is very close in space. So at 3 Tesla, when you have low resolution images, you may not be able to tell one part of the sulcus from the other bank of the sulcus. They are close in space. But in functional space, they can be miles apart. One part of the sulcus may be doing something very different than the other. And in fact, this sulcus, I will venture, although we have not specifically went and identified it, but just looking at its location, one part here could be the somatosensory cortex, and the other part, the primary motor cortex. It is well known that the sulcal banks can be very different in functionality. So these high resolutions are very important then for being able to obtain this, and we have the technology to do so. The other method that I described, which is very important for the Human Connectome project, is tractography. Tractography with many different orientations of data acquisition to be able to untangle fibers that cross. This is one version of it, what you are seeing. This is DSI, diffusion spectroscopic imaging, showing you a lot of tracks identified in the human brain, and it was obtained by these accelerated methods in about 8.5 minutes as opposed to normally 40 minutes that it would have taken. There are other flavors of tractography. For example, these are probabilistic tractography obtained with 7 Tesla, looking at connection between subcortical areas in the brain. For example, subthalamic nucleus and putamen and thalamus and other areas. These finger and arm-like looking things are essentially a probability surface within which there is a high probability of connection between nuclei in one case and the other case. And they can actually be correlated with resting state and functional networks as well. So we are looking at functional connectivity, if you like, with these resting state studies. And if you take a seed point, for example, in the brain over here, you can map actually how the other areas may or may not be correlated in terms of their basal oscillations in the brain. And you have also structural data that comes from the tractography imaging, which is a diffusion-based methodology. So if you put a seed point at the same place now, you also see structural connectivity to this point, and there are similarities and there are differences. If you put it on the frontal lobe, again, somewhat different pattern for this particular seeding point here in the frontal lobe. But again, between structural connectivity and functional connectivity, there are similarities and differences. And this is expected because functional connectivity can go through many, many different pathways, whereas structural connectivity is based on, if you like, hard wiring. So many similarities, many differences, and these are, of course, preliminary data in the Human Connectome project, and probably we can anticipate major improvements on that. But already we are very pleased with the progress of this particular project. And the idea is then ultimately, at some resolution, at some parcellation, to generate a connectivity matrix as shown here. So various different brain regions, you may parcellate it in terms of just using pure laboratory space coordinates. You may parcellate it based on task-based fMRI, but ultimately you can actually generate a matrix of connectivity either functionally or structurally between those regions. And the idea is, of course, in the Human Connectome project, this will be accomplished in the normal brains of approximately 1,200 people, identical twins, to generate a major database that can actually serve as a normal database for future studies on patients, development, etc. Now, I talked a lot about connectivity and functional, but of course, MRI, since its original discovery by Paul Lauterbur, has been used a lot for morphology and for clinical applications. But morphological imaging has also improved substantially beyond what is applied in clinical setting. I want to just show you towards this end of my lecture some examples. For example, vascular imaging. This is vascular imaging of the venous system. You can get beautiful detail of vasculature in using high fields compared to lower fields that are clinically available, like the 3 Tesla. This is work from my colleagues in Max Planck Institute in Tübingen. You can get exquisite anatomical detail of the cortex. So here are the gray matter areas in the cortex, but not only we are seeing the gray matter versus white matter differentiation, which has been known for quite a long time, but in these high resolution images and this particular contrast, this is unique to high fields, this phase imaging, you can actually see subcortical structures, subcortical myelination, for example, are easily detectable right over here. This dark line running right in the middle of the cortex in this striate cortex. Or subcortical nuclei or structures like the hippocampus with their intricate sections and subsections can be imaged right over here in a recent paper from our lab in radiology that impacts clinical imaging and clinical diagnosis. So there are many, many different areas in imaging, whether we are talking about functional imaging and whether we are talking about connectivity, whether we are talking about structural imaging, that MRI in the last 2 decades has improved significantly to actually change what we are able to learn about the brain compared to where we were 2 decades ago. And of course, it's interesting historically to note that nuclear magnetic resonance phenomena was discovered in 1945, and Ed Purcell from here, MIT, and Felix Bloch from Stanford actually received the Nobel Prize for that in 1952. And that particular activity for a long time led to NMR as a wonderful tool of physicists, but primarily chemists. But in 1973, then Paul Lauterbur primarily and Peter Mansfield has changed the game again using NMR methodology to create images. So they received the Nobel Prize in 2003 together, shared the Nobel Prize, and we launched into this fantastic period of being able to image human anatomy and later on function and connectivity and chemistry. And I can say that in 2011, new NMR technologies are still being discovered. And I have to add, take this opportunity to add that we really have to look at research in a very long period and very long time. So the fantastic useful outcomes of NMR were not planned. Ed Purcell did not foresee applications of imaging and indeed could not have been planned. And we have to keep this in mind as we go forward in research endeavors like this. And I think NMR and the development of it is an excellent example to look at as to how research impacts practical outcomes and what we can achieve with it for the human society. I'd like to end again acknowledging a lot of people who have contributed to this work, and you have seen their names in many of the slides, and I mentioned some of them as I went through. But most of the work comes from the Center for Magnetic Resonance Research, CMRR, where there's a team of people working on instrumentation, pulse programming, and image reconstruction, etc. And without that combined complementary work from all of these people, none of the work would have been possible. I would like to particularly mention several individuals like Isa Yacoub, Federico De Martino, Gregory Andriani, Gordon Shu, and Cheryl Allman, who have worked a lot on the functional imaging aspects of things. Amir Shmuel, who has worked on the decoding. My collaborators, Rainer Ugurbil and Jan Zimmermann from Maastricht on the MT imaging. The Human Connectome project, many individuals, but particularly Steve Smith, who has done the analysis for the resting state data, and Carla Miller from Oxford, and David Van Essen, my co-PI on the project. And I've showed you some images from Max Planck Institute in Tübingen from my colleagues and students there, Rolf Pohmann, Juliana Budde, and Denis Chaimov. With that, I'd like to thank you and like to end my talk at this stage. Thank you.
Could I ask the lights be turned on?
Yes, I have a question.
Yes.
The resting state network work originally, I mean, 10, 15 years ago, when we first saw it was dominated by 12-second rhythms, perhaps, something on that order. I wonder with the new fast and high speed and high-resolution imaging, whether you also find shorter cycling. And there was no time scale on your picture, so I couldn't tell.
Still, in the resting state networks, most of the biological signals that we are detecting are of the order of 0.1 hertz, which is about 10-second rhythm. However, they are not necessarily continuous, and they come and go. And I think that sometimes they come and go for very brief periods of time. And for that brief period of time, detection of those networks transiently becomes very important with these short TR acquisitions. So I think that people have realized that the transient nature of these oscillations. However, with these very long TR acquisitions, you may get 1 or 2 points, that actually deviates from that particular frequency, let's say 0.1 hertz. But on the other hand, 1 or 2 points on a looks like noise, and you are never sure to be able to extract yet another network from those 1 or 2 points. But now with these very fast acquisitions, those 1 or 2 points becomes 10, 20 points, and showing that they are really not noise, and you can actually then use that period, for example, to obtain information about what is happening to the network dynamics.
Any other questions from our audience? Don't be shy.
I think there's another one over here. Yeah.
Thanks, very nice lecture. Thank you
Thank
very much. So have you been able to, looking at the circuits in the brain, functional circuits, and relate them to some already known anatomical circuits, like the circuit of Papez, for example, or some of the basal ganglia circuits? Can you make a direct correlation between those images and some known circuits already known?
Yes. And in fact, the slide I showed you, the basal ganglia and the thalamus circuits, some of those circuits, those circuits in fact are known from the monkey work, and of course, in the human, they are not necessarily all mapped, have not been all mapped before. But on the other hand, those are some of the very first attempts actually showing those connectivity in the human. And you can do a reality check to a certain degree by analogy with the monkey. And there have been other examples of that, for example, from thalamic projections to cortical areas. There have been human data, again, from several labs, including ours, very similar to monkey data. So you can actually obtain, to a certain degree, verification, not in all cases, but comparing it with monkey connectivity, which is not obtained by DTI, but by invasive techniques. And I should add, that wasn't your question, but the basal ganglia work, for example, that is you can think of it as a basal ganglia thalamus connectome, but in this case it's actually for a practical reason, and those investigators from University of Minnesota are in particular interested in subcortical electrical stimulation for Parkinson's disease and things like that. And some of the nuclei, like the subthalamic nucleus, which is a target for them, is not very well visualized at 3 Tesla, but they are beautifully visualized at 7 Tesla. And it gives us the ability to actually look at the connections and the functional connectivity and anatomic connectivity in order to understand a little bit more about what those nuclei may be doing and combine it with that electrophysiology data that comes from patients.
Can you say a few words about the ultimate limits, of physical limits, in terms of how high the field strength can go? What other factors will limit the resolution that is eventually possible?
I actually see within current magnetic fields, we will probably, I showed you images of, 0.75 isotropic, and I have to emphasize again, those are, if you like, multi-slice, each slice a single-shot EPI image. So they are the whole brain I showed you acquired in 6 seconds, but now around, let's say, a second or sub-seconds. But these are very fast imaging, so you can, if there's an application where you can time average, take more time, you can get somewhat better resolution. But I think that within current technology, with not a new discovery, but improvements and implementations, we will probably see half a millimeter or so isotropic resolution images in the human brain for this kind of fast acquisitions. And there are efforts to go to higher fields. We are expecting a system that will operate at 10.5 Tesla. And that's a whole body system, and there is a brain-only system that is being installed at NIH Intramural Research for 11.7 Tesla. And there are plans in France for going to 11.7 Tesla for whole body imaging. And you can see that 10.5, 11.7, there is a reason for that. To go beyond that, then the magnet technology changes quite a bit. So you cannot use the same wire anymore for making those magnets. And you get into technology which is extremely expensive, wires that are extremely expensive and hard to work with, higher failure rates, etc. So we are then going into, instead of magnets costing, you know, several $1 million, into a new regime of very large magnets that are extremely expensive. And I think before we go there, we will have to digest 10.5 Tesla and 11.7 Tesla. So around 12 Tesla, the wires fail. So that is a limit that we are going to live with for a while, although in South Korea, there is an effort to go to 14 Tesla. But it's a huge, huge project with extremely expensive project, and they may be able to put the funding together to do it. Ultimately, we have physiological limits, health limits that we have to worry about, and certainly at 7 Tesla and 9.4 Tesla, where there's quite a bit of experience now, these health limits, to our knowledge, over thousands of, several thousand people examined, don't quite exist. And will they be there at 14 Tesla, 16 Tesla? Probably not, because we do experiments with animal models at 16 Tesla in a small bore magnet. And of course, we don't really know what happens to their cognition. However, at least their hearts and livers and circulatory system, etc., works fine. So there are practical limits based on cost and physiological limits to worry about as well.
If you could get a whole brain image at the scale of individual neurons, do you think you could explain the phenomena of human consciousness?
I think people make too much about human consciousness. I don't think I need individual neurons. I mean, not me. I don't think the field needs individual neurons. And so we first have to agree what human consciousness is, however. So that's a whole new discussion, and we can stay here and discuss that. But of course, well, I will say a little bit, human consciousness can be a very interesting phenomena that somehow are linked to maybe old historical concepts that there's something unique about human consciousness, but it may not be that unique. It may be a simple somehow processing of our memories combined with sampling of our inputs and actions, and that latter concept is not very far from being understood in the human brain. Not fully understood for sure in a mechanistic sense, but we are getting close to it, I think.
Okay.
And I certainly believe you don't need individual neuron connections.
Okay, I think I have to put on my program chair hat, and thank you so much for a wonderful talk. Your comment about many new challenges still waiting to be solved is inspiring. And so on that note, we thank you very much for a very inspiring talk. Thank you.